Okay, very good afternoon, everyone, and thank you so much for tuning in for today's webinar. Today, I will be sharing about customized protein production and antibody generation for diagnostics development and manufacturing. For those of you who have heard of GenScript for the first time, I would just like to give a very brief introduction of the GenScript group. GenScript is founded in New Jersey, US in 2002, which is 20 years ago. We launched the first custom gene synthesis platform, and as a natural extension of the gene synthesis, after 2 years, we established our custom protein and antibody services. And in 2015, we are listed in the Hong Kong stock market. And in last year, we opened a new R&D and production facility for recombinant protein in Singapore. The GenScript group have 4 different subsidiaries, which have focused on different applications. The GenScript Life Science Group is a life science CRO for research services, where the Bestzyme is an industrial enzyme producer, and the Legend Biotech focus on cell therapy, and the GenScript ProBio is a biologics CDMO. For the GenScript Life Science Group, we provide services including oligonucleotide synthesis, gene synthesis, peptide synthesis, recombinant protein services, as well as reagent antibody development. And for today's talk, I will be mainly focused on the recombinant protein services and also the reagent antibody development. So for today's webinar, first I will give an overview of the IVD industry, and following that, I will be covering the different IVD techniques and the key bioactive raw material that is key determinant of the assay quality. As a further illustration of the immunodiagnostics, I will use COVID as a case study. Well, for immunohistochemistry, I will use the breast cancer diagnosis as a case study. And lastly, I will also share with you about the GenScript services for IVD kit development and manufacturing, including our oligosynthesis, recombinant protein, and antibody generation, with the main focus on the monoclonal antibodies. So the IVDs are medical devices used to perform diagnostic tests in vitro on biological samples. It helps to detect and monitor diseases and influences approximately 70% of clinical decisions. The global IVD market is forecasted to grow from 70 billion in 2020 to 107 billion in 2029, with an annual growth rate of 6%. When we look at the North America IVD market size, we see these 2 outstanding bars, which is a result of the COVID. The COVID has surged the IVD demand, lead to a growth of 25% in 2020 versus the previous year. It has, with the widespread use of the antigen rapid test, it has also increased the demand and adoption of point-of-care tests as a whole. And the key driving factor for growth of the IVD market is the rising prevalence of chronic and infectious disease worldwide. So next, I will discuss the different players in the IVD industry. The upstream arm of the IVD industry is the raw material providers, and GenScript position ourselves as a reliable partner to provide the key bioactive materials such as the antibodies, antigen, and enzyme. And the midstream of the IVD industry are the product owners who perform the product development and commercialization. While the downstream arm of the industry are the end users, who are the hospital lab or clinical lab if it's a lab test, or if it's a point-of-care test, the users will be the layman who will use it at home, or the clinicians use it at the physician's office, or other low-resource settings. In terms of the IVD techniques, the most commonly used technique include the clinical chemistry, molecular diagnostics, immunodiagnostics, microbiology, and hematology. For today's webinar, we will be covering the clinical chemistry, molecular diagnostics, and immunodiagnostics. Clinical chemistry involves the use of simple chemical or enzymatic reaction for detecting aniline in blood or urine. Here, I would like to illustrate this technique with glucose detection. So, as shown in this reaction formula here, the detection of glucose is based on the enzymatic reaction using glucose oxidase. So, the image shown here is a glucose detection in urine, and once the enzymes are coated on the strip, and once you dip the strip in the urine, the color there will be change in the color that is indicative of the amount of the glucose present in the urine. And some other routine tests are also based on the enzymatic reaction. For instance, the detection of creatinine and blood urine nitrogen for determining of the kidney function, and also the detection of the total cholesterol and triglycerides for the lipid panel. For the clinical chemistry that is based on the enzymatic reaction, the enzymes are the key bioactive material for such tests. And the key requirement for such enzyme is that they should have good stability and very high activity, so that the test is sensitive enough to detect the amount of the aniline. And the manufacturing method for the enzymes, for most of the enzyme is recombinant protein expression in the E. coli system. So next, moving on to molecular diagnostics. Molecular diagnostics involves the use of molecular biology technique for diagnosing diseases. And the most widely used molecular diagnostic method is the qPCR and qRT-PCR, which involves the amplification and detection of DNA and RNA sequences present in the sample. As illustrated in this image here, the RNA is first isolated from the sample and undergoes a process called the reverse transcription to obtain the cDNA, after which the cDNA are subjected to the qPCR process for the detection of the amount of the cDNA present in the sample. Some of the key trends in the molecular diagnostics is the detection of circulating microRNA for cancer diagnostics. MicroRNA are shed by cancer cells, and they are extremely stable because they are protected by the microvesicle or the RNA binding proteins. Therefore, they are considered as a very good biomarker for cancer diagnosis. And another key trend in this area is the increasing demand in non-invasive prenatal testing, which aims to prevent giving birth to children who have genetic disorder by detecting such genetic disorder at the early stage. Such test is based on the cell-free DNA, which is from the fetus that is released to the mother's circulating system. The technique usually used is the gene sequencing to sequence the cell-free DNA in the mother's blood to determine if there is any genetic disorder in the fetus. For the qRT-PCR, the key bioactive material used include the primers and probes. And primers are probes. It is very crucial to have high purity and the stability to avoid cross-contamination. And the manufacturing method for primers and probes is oligosynthesis. And for the reverse transcriptase and DNA polymerase used in such process, it is also required for them to have very good stability and activity. And for such enzymes, they are usually produced by recombinant protein expression in E. coli system. So next, moving on to immunodiagnostics. immunoassays are detection of analytes using specific antigen and antibody reaction. So if we are aimed to detect certain antigen, we could use the antibody to detect the antigen and vice versa. For the immunodiagnostics, there are three main modalities, and the most high throughput and automatable technique is the ELISA or CLIA-based test. So such tests are usually performed in the hospital lab or clinical lab to identify the amount of certain analytes. And the second modality is the lateral flow assay, which is a point of care testing. For instance, the COVID rapid antigen test or pregnancy test. Such tests are for home use, and they are usually very low cost. And such type of test is usually used for detecting the presence of the analyte rather than the quantification of the analyte, meaning that such tests will usually show a positive or negative result, where the ELISA or CLIA will show the amount or the quantity of the analyte. The third modality is the immunohistochemistry. Such tests are usually performed in the hospital lab for biopsy tissue. For instance, the breast cancer biopsy tissue. And this type of test is used for detecting, is used for analyzing the presence as well as the distribution of the analyte to detect which region, which tissue is expressing such biomarkers. The key bioactive material used for immunodiagnostics are the antigen and antibodies. For such material, it is very crucial for them to have high sensitivity and specificity against the target. And the manufacturing method for antigen is the recombinant protein product expression. It can be either in the E. coli system or mammalian platform, depending on the complexity of the protein. And for antibody, it depends on whether the antibody's amino acid sequence is known or not. If it's a known sequence, we can opt for the recombinant protein expression in a mammalian system. And if it's a novel target and the sequence is unknown, we will need to perform the in vivo antibody generation using animals. So the in vivo antibody generation utilize the animal's natural immune system to generate high quality antibody lead. So next, I will go through the workflow of such in vivo and of this type of in vivo antibody generation process. The first step is to optimize the antigen and produce the antigen. The antigen can be a protein, or it can also be a peptide. So after we obtain the optimal antigen, we will inject it into the animal. It can be rabbit or rodents. And among the immunization of the antigen, the animal will produce the antibody. And following that, we will perform the extraction and the purification of the antibody. So the antibody is categorized into the polyclonal antibody and monoclonal antibody. Polyclonal antibodies binds to multiple epitopes on the antigen, while monoclonal antibody only binds to one single epitope on the antigen. At GenScript, we provide a one-stop solution for all custom antibody needs, including rabbit or rodents, and also polyclonal or monoclonal antibodies. So next, to further illustrate the immunodiagnostic technique and also the immunohistochemistry technique, I will discuss further using COVID and breast cancer diagnosis as an example. So for COVID, during the acute phase, what is detectable is the viral RNA and also the viral protein. And usually after 2 to 4 weeks after the infection or vaccination, the body will start to generate the adaptive immunity response. which will generate the antibody against the virus. So during the acute phase, it is usually recommended to perform the detection for the viral RNA, which is based on the qRT-PCR or the viral protein. And during the later phase of the infection, the antigens are not detectable anymore. Therefore, we will need to detect the antibody to determine if the person has a past infection or to determine whether the person has generated immunity against this virus. For the nucleic acid detection, it detects the RNA in the virus, and while for the viral protein, we usually detect the M protein. While for the antibody detection, it depends on the type of application. We can be detecting the IgG, IgM, total antibody, or the functional antibody, such as a neutralizing antibody. For the antigen detection, the most commonly used are two modalities, including the LFA or the ELISA. For the LFA, is also known as the antigen rapid test. So for such tests, it is required to have a pair of antibody that is specific to the M protein. So the first anti-M protein is coated on the conjugation pad with a nanoparticle for labeling. So once the sample is loaded on the sample pad, you will bind to the antibody and also become labeled. And afterwards, you will continue to flow to the test line, which is coated with also another anti-N antibody to capture the complexes. Therefore, you will give a colored line indicating that the M protein is present in this sample. While for the ELISA, it also operates in a very similar manner with a capture antibody that is coated on the microplate to capture the M protein, and also the detection antibody that will sandwich the M protein and also give rise to a colored signal. For the antibody detection, which is also known as the serology test, I will illustrate further with the IgG ELISA kit and also neutralizing antibody ELISA kit. For the IgG ELISA detection, the antigen, which is the spike protein, is coated on the microplate, and the IgG and the antibody that is specific to the spike protein will be captured. Following that, the anti-human IgG antibody labeled with the HRP will detect the IgG antibodies and to also give rise to a color signal. Another type of ELISA kit is the neutralizing antibody test. So such test detects the functional antibody that can block the virus interaction with the human cells. As illustrated in this image here, the virus enters the human cells through the binding of the spike protein to the ACE2 receptor. And if there is neutralizing antibody present in the serum, it will block the binding of the spike protein to the ACE2 receptor. So by mimicking this process in the ELISA format, using the purified spike protein and ACE2 receptor, we can detect the neutralizing antibody using a much simplified method based on the ELISA platform. So by conjugating the spike protein with the HRP, if there is neutralizing antibody present in the serum, it will bind to this region and block its binding to the ACE2 receptor. So next, I will go deeper into the immunohistochemistry, which is a technique that is commonly used for breast cancer diagnosis. The immunohistochemistry is selectively imaging antigens in cells of tissue selection based on the principle of antibodies binding specifically to antigens in biological tissues. IHC can be based on the detection can be based on chromogenic or also can be based on fluorescent. So for chromogenic detection, the tissue is first fixed, and the primary antibody is used to detect the antigen, where the secondary antibody is usually conjugated with the peroxidase for amplifying the signal and give to a colored product. So the image here shows the staining result using such method with the rabbit beta-actin monoclonal antibody, which shows a very specific detection of the beta-actin. For the fluorescence detection, the only change is what is conjugated on the secondary antibody is a fluorophore instead of the peroxidized or alkali phosphate. For the fluorescent detection, here is a comparison between the mouse monoclonal antibody and the rabbit monoclonal antibody, and we can see that there is a significantly higher binding of the rabbit antibody as compared to the mouse counterpart. So this shows that the rabbit monoclonal antibody is particularly suited for the IHC applications. And in the merged image, we can also see that the rabbit antibody is able to detect a certain region that is not detectable by the mouse antibody. So further to illustrate using the breast cancer diagnosis using the IHC. So the 4 different markers that can be stained using the IHC include the estrogen receptor, progesterone receptor, HER2, and p53. For the estrogen receptor, it binds to the hormone estrogen, while the progesterone receptor binds to the hormone progesterone. So for the 2 hormones, they are, because of the overexpression of this receptor, it leads to the overproliferation and the overgrowth of the cancer cells, and leads to the which means that such type of expression indicates that the endocrine therapy are the better therapy to for the treatment. Where for the HER2, HER2 is an oncogene, and as shown in this image here, if there's an overexpression of the HER2 gene, there will be overexpression of the HER2 receptor on the cell membrane, which will lead to the overproliferation of the cells and also the cancerous tissue. So if the staining of the HER2 reveals that the expression is very high, it means that the malignancy of the tumor is also higher, and the suitable therapy for such case will be the anti-HER2 therapy, such as the monoclonal antibody Herceptin, which will block the HER2 receptors. Another marker that for detecting of the breast cancer is the p53, which is a tumor suppressor gene. And high expression of the mutant p53 indicates that the cells have very high proliferation and higher degree of malignancy, and also associated with poor prognosis. So in this study here, it was shown that the rabbit monoclonal antibody have a higher sensitivity than mouse monoclonal antibody for estrogen and the progesterone receptor in breast cancer using the IHC method. And here is the FDA approved IHC diagnostic kits for detection of the HER2, which are also both using the rabbit monoclonal antibodies. So next, moving on, I would like to introduce our services for IVD kit development and manufacturing. So for those of you who are working on the molecular diagnostics, such as qPCR or RT-PCR, we do provide the service for synthesis of the probes and primers, and our purification process is developed and optimized for diagnostic purpose with a very high purity. So some of the advantages from us is the high purity, which means that there's very low exogenous contamination, and also the fluorophore signal from our probes are very stable and give rise to very consistent fluorescence signal. And we also have very high batch-to-batch consistency. So next, I would like to give an overview of our protein expression service. So for the recombinant protein production, what we require is the amino acid sequence. After that, we will perform the codon optimization, gene synthesis, and construct generation. And after we obtain the construct, you will be subcloned into the cells, for obtaining the protein. And after which we'll perform the purification and characterization of the protein. For the cells that use for producing the protein, we have the bacteria, insect, and mammalian platform. And usually for enzyme and antigen, we can opt for the E. coli expression system. And usually if it's antibody, we can opt for the mammalian expression system. For the E. coli expression system, is optimized for the enzyme manufacturing. So for the it started with the plasmid design and construction, moving on to fermentation process, which we have up to the scale of 500 liter, and following that is the purification and analysis of the enzyme obtained, and lastly we can also perform the neutralization for the enzyme, and also followed by the QC methods to ensure that the quality of the enzyme produced is up to the standards of the intended application. So if it's an enzyme for IVD enzymatic reaction, for instance, the glucose reaction, we can perform the assay. We can perform the chemical enzymatic assay that is catalyzed by the enzyme produced to ensure that its activity is high enough and working well. And if it's an enzyme used in the molecular diagnostics, such as the DNA polymerase, we will make sure that it has the complete removal of the nuclease using the fluorescence detection method. And also some other enzymes, such as restriction endonuclease for the cloning process, we can also perform the functional QC test to make sure that it does cleave the intended restriction sites needed. Next, moving on to the mammalian expression system. This is especially useful for those who are working on antigen detection and require the production of recombinant antibodies. Here, I would like to introduce our TurboCHO platform, which is our preparatory platform that is optimized for recombinant antibody production. The TurboCHO platform can have different modalities. So if you require the testing of multiple sequences at a lower scale, you can opt for the high throughput package. And if you have already identified the sequence and require higher amount of less than 500 milligram, you can opt for the CHO Express. And if you have already entered the manufacture phase, where you need large quantity of very consistent antibodies, we also have the stable cell line that is suitable for such application. So for the stable CHO services, it is good for high quantity that is at a gram level. So how can the TurboCHO platform fit into the IVD pipeline? As I mentioned just now, so if you are at the development and optimization stage where you require testing of multiple sequences to identify the best candidate, you can opt for the high throughput services, which we can deliver the final antibody in as short as 2 weeks. And if you are in the phase of assay validation, you can opt for the CHO Express service for the higher amount. And if you are already in the manufacturer phase, you can opt for the stable cell line, the CHO High Performance, which can reach up to the yield of 3 grams per liter. So here I would also like to take this opportunity to let you know that we have newly launched our TurboCHO platform with a brand new manufacturing facility in Singapore. And the turnaround time from gene to protein or antibody is as fast as 2 weeks, and starting from USD 399 per sample. So in the last part of the webinar, I would like to go through our antibody generation services. So this is we have GenScript. We have the state-of-the-art animal facility, which have the ALEC International and OLO certification, and we provide a broad range of antibody services for individual or bulk orders. So for the mouse monoclonal antibody generation, it requires the generation of hybridoma. So as I mentioned earlier on, the process starts from the antigen optimization and production, followed by the immunization of the rodent. And after that, the antibody producing B cells will be fused with myeloma cancer cells for immortalization to generate the hybridoma cell line that can indefinitely produce the antibody. So for this service, we'll provide the hybridoma cell line, which can provide an unlimited supply of antibodies by just growing them. And with our proprietary monoboost technique, we are able to achieve a very high percentage of positive clones from one animal. As shown in this 2 case study here, when we use the monoboost technique, which is optimized to induce the higher immune reaction from the rodent, we are able to achieve more than 90% positive clones. And next, I'm going to introduce the MonoRab, which is our customized rabbit monoclonal antibody production. As we know, the rabbit has a more robust and unique immune system that enables the generation of antibodies of a very superior specificity, affinity, and diversity compared to rodents. And the underlying reason of this include the stronger immune response of the rabbit, and also the longer evolution distance, and also the higher genetic heterogeneity of the rabbits. So in this case study over here, both the anti-sera from the mouse or rabbit are compared. So we can see that in the mouse sera, we see the band, the amount of the band is much more than the rodent sera. So this means that it has a much higher diversity of the antibodies compared to rodents. So to further illustrate the process of the MonoRab generation workflow, it first starts with the immunization of the rabbit, followed by the isolation of the PBMC, and enrichment and expansion of the antigen positive B cells. After that, the B cells are subjected to screening and supernatant harvest. And after identifying the positive antibodies, we will perform the sequencing of the B cells and perform a small-scale transfection to give rise to recombinant antibody. So the advantage of this process is that it is fusion-free, meaning that in this process we do not need to generate the hybridoma, and so this will avoid the B cell loss due to the efficiency of the hybridoma generation. And this process is also fax-free, so you will give rise to better B cell viability, and this will also result in a much larger cell pool with around 100,000 memory B cells seeded and screened. So for this package, it takes around 13 weeks, and we also have an express package available with the beacon screening system that can shorten the timeline to around 10 weeks. So next, I would like to give a case study using the SARS-CoV-2 monoclonal antibodies generated using the mono-rab system. So in this project, the antigen we use is the extracellular domain of the spike protein, and for this, using the mono-rab platform, we are able to discover 16 monoclonal antibody across 11 different epitope bins, as shown here. So when we test the antibody-antigen affinity analysis using a BR core system, we found that the affinity of the antibody generated is very high. So using the mono-rab platform, if your application needs to have antibodies that have very high epitope diversity and high affinity, the mono-rab could be a suitable service for you. So next, I would like to share our track record in using our protein and antibody services for immunodiagnostics. So in 2020, we have produced the key raw material and co-invented ELISA kit with Prof. Wang Linfa in Singapore, which is the CPAS SARS-CoV-2 neutralizing antibody test, which I have also earlier introduced briefly as a case study for the immunodiagnostics. So the CPAS neutralizing antibody test has obtained the HSA authorization in Singapore within 70 days from its ideation. And after a few months, it received the FDA EUA authorization, which makes it the first FDA authorized test for neutralizing antibody detection. Till today, this test is widely used in 80 countries, and it has registered for in vitro diagnostic use in more than 40 countries. For this test, the key bioactive raw materials are produced by us, including the antigen and protein expression in our mammalian platform, as well as the in vivo antibody generation. So to a quick summary of today's webinar. So we know that the bioactive material, including the antigen, antibody, and enzyme, are very important determinants of the quality and performance of IVD assay. And GenScript E. coli platform is optimized for enzyme manufacturing and scale up to kilogram level. And our TurboCHO platform is also optimized for the production of the recombinant antibody within a very short time frame, as fast as 2 weeks. And also we provide a one-stop solution for in vivo antibody generation services using both the rodent and rabbit system. And our mono-rab, which is the rabbit monoclonal antibody services, can generate the monoclonal antibody with very high affinity, specificity, and diversity, which is suitable for tissue-based diagnostics. With that, I would like to end today's webinar, and thank you for your attention. So if you have any question, please feel free to ask me. Yeah, thank you very much.